All right, everybody. I'm turning on the webcam. I hope that it works for us. There we go. That's it. Okay, that is my mug. I will show your mugs in just a second. We are going to go ahead and do our third review. This is the heredity and gene expression, so units 5 and 6. Oops, I'm doing all the wrong things. Slideshow is what we need to do. There we go. And let's see if we can show who's here. Wave, everybody. Who's there? Am I showing everybody? There we go. Did I get you on the screen? Can you see yourselves? Okay, and then over here, I am just so. Are you in the image? I am just so impressed with the people who are here on Friday afternoon. Oh my gosh! But we are recording it for those people that are not here. So let's go ahead and get started. I am going to talk as fast as my big mouth. I always think I have kind of a big mouth. How fast my big mouth can go. So the first thing we're going to talk about is meiosis. Remember that we called mitosis non-reductive. And meiosis is called reductive, and that's because you're reducing the number of chromosomes that are in the cell. So mitosis, you start with 46 chromosomes in a human cell and you end up with 46 chromosomes in a human cell. With meiosis, you start with 46 chromosomes in a human cell, and how many do you end up with? 23, half of the DNA. So it is reductive eukaryotic. It creates haploid. Why am I emphasizing the half the DNA? So if we have a plant cell with 20 chromosomes, how many chromosomes are in the pollen? 10, always half, okay? Sperm, egg, or pollen. Oops, I'm going to make this little menu go away. The diploid cell that forms when two gametes come together is called a zygote. A zygote is the tiniest of the babies. When sperm and egg or pollen and egg come together, that is a zygote, and it will start dividing through mitosis to form us a bambino. What did I just press? Did I just trigger something? It's gonna screw up my recording. I'm trying to get it to go and nothing is happening. I'm like, there we go. Okay. Every time I have troubles with this. All right, let's just review really quickly chromosomes in a diploid cell. What does the prefix DI mean? Two. It's a different language than BI, and I never remember whether it's Greek or Latin, which one's which, but DI means two. So two copies of each chromosome in the cell. So you get one from biological mom and one from biological father, and they're called homologous pairs or homologs. That's kind of a fancier word than homologous pairs. Don't confuse homologous pairs with sister chromatids. So take a look at the cartoon on the left. Notice that one of the chromosomes is this way, but it's got two sides to it and it's leaning to the side. The two sides are the sister chromatids. The other one is leaning this way and it has two sides. The two sides are the sister chromatids, but the one that's leaning this way and the one that's leaning this way, those are the homologous chromosomes. I think that's kind of confusing. So two chromosomes, each of those has their identical copy attached. Remember that sister chromatids are identical copies, so the left side and the right side are identical to each other. Okay, so what stage of mitosis are we looking at? That is prophase, because we know that it's got the full set of DNA and they're all bivalent. They all have sister chromatids, so we know that that's prophase. Meiosis and mitosis look a lot alike. So over on the right side, the blue area is what mitosis is. It's fewer steps. And then the yellow side is meiosis. So it says instead of IPMAT, interphase, prophase, metaphase, anaphase, telophase, in meiosis we have IPMAT, PMAT, because it divides twice. So the first half of meiosis is denoted with a Roman numeral 1, and then the second half is denoted with a Roman numeral 2. So like prophase 1 and then prophase 2 in the second half. The chromosomes cross over. That's the only time that chromosomes cross over is during meiosis. Doesn't happen during mitosis, and that is a very important concept. So that's going to create new genetic combinations. So mom's chromosome crosses over with biological dad's chromosome. They cross over and create new chromosomes that have never existed. The way they do that is lining up side by side. So look in the yellow area. Do you see where it says crossing over right here? They're crossing over. I wish I could make that bigger for you to see. And so this is where the chromosome number gets cut in half, is right here. When the cell divides, half the DNA goes to the left cell and half the DNA goes to the right cell. And then we just need to split the bivalent partners from each other at the end. The second half of meiosis is denoted with a Roman numeral 2. It is identical to mitosis, but the chromosome number is half. So if this were in a human, we would have 46 bivalent chromosomes that are side by side, 23 pairs. And then now I would have 23 bivalent in this cell and 23 bivalent in that cell. Samby, you need to go get a drink of water. I see you just. I know. Go get a drink of water. That's how you do it. You should be eating something. You should have a piece of candy in your mouth, something. All right. Prophase 1. This is meiosis. Prophase 1. The homologous pairs are connected to each other in synapsis, and they form structures called tetrads. The prefix tetra means four. At each connection, called a chiasma, you don't need to come up with these words on your own, by the way. I'm showing you these words because you might read them in a question, and I don't want you to be like, oh, never seen that word before, panic. Just read the question. Don't worry about the vocabulary so much and you'll get the hang of it. So at each connection, called a chiasma, DNA is exchanged or swapped between the homologous pairs. That's what crossing over is doing, and it's creating unique chromosomes, a cross between the mom and the dad. And again, what are those four cells at the end of the diagram? What is the arrow pointing at? Gametes is a great word for it. What else are they? Eggs, sperm, or pollen. Good. And they are all haploid cells because they have half the DNA. The first cell up at the top was diploid. They're all diploid, 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 diploid until the cell divides. And now these cells are all haploid because they have half of the DNA. Okay, and then just understanding what genetic recombination is. We're going to use that word a few times. Crossing over is an example of genetic recombination. We have a new combination of the DNA of the genetic material. Mutations increases or is what we call genetic recombination. We get a mistake during the S phase, and the DNA now is different. So that's genetic recombination. And then random fertilization. We don't know which sperm or pollen and which egg are going to come together. So when they combine, that's genetic recombination because we're leading to a new organism that is unique, something that hasn't existed before. So genetic recombination is kind of the opposite of what cloning is. Cloning doesn't do these things, although cloning does have mutations. Okay, moving on. Mendel's law of segregation. Remind me really quickly from history class, what does segregate mean? To separate. To separate. So only one of the two alleles for each trait goes into a gamete. So you have like 20 to 25,000 genes. Half of them go into one sperm or egg, and half of them go into the other sperm or egg. So segregation means separation. The alignment at the metaphase 1 plate is random. So whether the little t is on the right or the left is random. And you have 20 to 25,000 of those genes that are lining up randomly. So any individual, any sperm or any egg is going to be super, super unique. So which side each partner chromosome lines up on is random. And then Mendel's law of independent assortment. Separate alleles for separate traits are passed on independently of each other. You don't need to memorize this necessarily, but as long as the alleles are unlinked. So what they're saying is that chromosome A and chromosome B get passed on separately. If the gene happens to be, if they're both on chromosome A, then it's not separate, then it's not independently assorted. So it says to the right, when Mendel looked at two different traits, were they inherited together or independently? So we've got the trait for the color of the pea plant. It could be either yellow or green. And we've got the trait for the shape of the pea. Sorry, it's the pea. It could be round or wrinkled. I've got some silly boys in the front row here. So if we take a look here, using the Punnett squares, the prediction, if they are dependent, if they're inherited together, we should see a 3-to-1 ratio in the offspring. But if they are inherited independently, then we should see a 9-to-3 to 3-to-1 ratio. Yesterday I couldn't, yeah, I can't move my image around. I'm supposed to be able to drag it around, and for some reason I can't. So the results, there were 315 yellow round, 108 yellow green, that didn't make sense, green round, yellow wrinkled, and green wrinkled. That's a 9-to-3 to 3-to-1 ratio. So that means that the correct answer for this is that they were inherited independently. And for some reason, that's not working. There we go. You can't see it. Okay, Punnett squares, just a real quick reminder that when you have a dihybrid problem, and the problem is behind my video, I'm really sorry about that. You have a dihybrid problem, so it's like Y's and R's. You could make that big Punnett square, or you could just make two little Punnett squares and multiply the probabilities. And so I gave you some problems in this unit where we had like 5 traits, and you had to figure out, so that Punnett square would be enormous. It's a lot easier just to do 5 Punnett squares, 1 Punnett square for each trait, and then multiply the probability. So watch how we did this. You have 2 traits, the color and the shape of the P, and then you multiply the probabilities. What are the odds that they would have a yellow round P? You take yellow from the left one and round from the right one. 3/4 times 3/4 is 9/16. Okay, moving on. The test cross. The test cross is when you have a dominant organism, an organism that's showing the dominant trait. Nick, come on, you guys, focus. When you're showing an organism with the dominant trait, so like purple in this example, you can't tell if the purple is homozygous dominant or heterozygous. There's no way to know that. So the only way to find out for sure is to mate it with a what? Does anybody know? With a recessive. Very good. So mate the organism with a recessive phenotype. If half of the offspring are white, then you know it was heterozygous. If 100% of the offspring are purple and there are no white, then you know that it was a homozygous dominant individual. So that's what a test cross is. Okay, and then just a quick reminder about codominance and incomplete dominance. Codominant means they're both dominant. So the example was blood types A and B. If you get an A allele from one parent and a B allele from another, you're going to have blood type AB. They both show up. Incomplete dominance is where they blend. So if you get a dominant gene from one parent and a dominant gene from the other, they blend. So the example in this one, red is dominant and white is dominant. And if you get a red gene from one parent and a white gene from the other parent, what color is the flower? Pink, because it is blending. It is not a red and white flower. Okay, and then polygenic inheritance. It's so easy to mix up some of these words. What does poly mean? Many, and then gene. So this means that many genes are involved in determining a particular characteristic, like skin color, eye color, height. Those are some of the classic ones. And truthfully, they think almost all of our characteristics are in fact polygenic. There's very few characteristics that are determined by one gene and one gene alone. An example of a characteristic that is determined by one gene is blood type. Your blood type is determined by one gene. But most of the other things that we have in our bodies or the way we look at ourselves or whatever is many genes working together. Okay. Sex linkage, just to remind you, if the trait is carried on one of the sex chromosomes, and it gets complicated because not all organisms have the same sex chromosomes. So in human beings, 2 X's is a biological female, and X and a Y is a biological male. If it's carried on the X chromosome, then the female is going to get 2 copies of it and the male is going to get 1 copy of it. What is the advantage in that circumstance to the female XX? They have a backup. So if one of the X chromosomes has, say, red-green color blindness or Duchenne's muscular dystrophy or hemophilia, they have a second X chromosome that might protect them. Not that red-green color blindness is something we need to be protected from, but so it's an advantage. So we see less sex-linked characteristics in females than we do in males. Males automatically have that trait, whereas females, they have to get usually 2 copies of it in order to exhibit the trait, in order to actually show the trait. Okay, and then just remember how you do a sex-linked pedigree, that you show the dominant or recessive trait as a superscript. Notice that the Y isn't carrying it, so leave the Y blank if it's carried on the X chromosome. Who is the only one that gets the Y-linked traits? Males again. So the females are not going to get those traits either. Okay, so it says some genes are inherited together because they are found on the same chromosome, and that's called linked. This is gene linkage that we're talking about. So if they're on the same chromosome, so example in the diagram there, A, B, and C are inherited together because they're on the same chromosome. Crossing over can separate them, but they're on the same chromosome. So we're going to build 2 things here. We're going to talk about mapping. Do you remember chromosome maps? And we're going to talk about how we determine what those chromosome maps are based on whether they crossed over or not. Okay, so it says if the genes are on different chromosomes and inherited separately, then that's the dihybrid problem we would have. And this is what we would predict for the offspring. So if the example here is the body color, gray or black, versus the wing shape, vestigial, which means like little tiny wrinkled wings, versus the full wings. So those are the 2 choices. Usually when you have a plus, that means wild type. So that's the sort of most common type. And then the without the plus would be like a mutant version of it. So they don't have the other trait. Okay, so in this test cross, we're predicting that we would get the same number of gray full wings, black vestigial wings, gray vestigial wings, and black full wings, that we would get equal numbers of them. However, give me a second. So if genes are located on different chromosomes, we get a 1-to-1 to 1-to-1 ratio. I'm continuing, I'm not there yet. If the genes are linked on the same chromosome, then it's just a monohybrid problem, and we put it all together in one box. You had 2 Punnett squares last time, now you have 1 Punnett square, and the prediction is that half of them will be gray normal wings and half of them will be black vestigial wings. So this is what the ratio should look like if they're on the same chromosome. So I don't know if you guys remember this, but when we do the actual cross, what we get isn't any of them. The results, I'm in the way, but we have 955 to 944, and then 206 and like 206 again, or something like that. What is it? 185. Are you looking at it? Thank you. That's super helpful. Okay, so that's not either of the predictions. That's not 1-to-1 to 1-to-1, and that's not 1-to-1, 0-to-0. So who remembers what actually happened in this circumstance? Why are there so many of the black, let's see, of the gray normal and black vestigial, but then there's a few of the others? What actually happened? Crossing over. Okay, they are on the same chromosome, they are linked, but they occasionally get separated from each other if they're separated during a crossing over event. So it says here with chromosome maps, we'll come back to it in just a second, we can use the recombination frequencies to make a linkage map. Genes that are further apart will recombine, cross over, more often than genes that are closer together. A map unit, this is something that you could, maybe have to do real basic math on. A map unit is the distance between genes on a linkage map. So in the example, one well, this is not the example, this is always the way it is. One map unit equals 1% chance of crossing over and recombinant offspring. So in the diagram, the color and wing genes are separated approximately 17% of the time. So we place them 17 map units apart. But apparently, this gene here separates from this gene less often. And so we put it closer to that gene. It separates about 9% of the time. And does anybody remember? This is so obscure. Why does this, look at this 9 and 9.5% versus 7? They don't add up. Who remembers why? Sammy? Oh, listen to you guys. Very good. Double crossing over is exactly right. So that screws up the statistics a little bit if they cross over twice. So not important. Maybe it would show up. I don't know. All right, let's remind ourselves just about the pedigrees. Again, pedigrees are always, when they're not looking at gender, they're looking at biological. So this is like the sex assigned at birth. So we're showing unaffected males as a uncolored in blue box in this case, and unaffected females as an uncolored in blue circle. And then if they have the genetic trait that we're studying, it may or may not be a good trait or a bad trait. It doesn't matter. They're colored in. So part of what we needed to do is analyze these. So it says, is the red genetic trait due to a dominant or recessive allele, or are both possible? So let's take a look and see if we can figure it out. So this female has it. 3 of her children have it, and 2 of her children do not have it. If it's recessive, what do we know about this male? What? He's hetero, and therefore that's how the children have it. Could it be a dominant trait? So then mom could be heterozygous or homozygous. She could be heterozygous. So let's think about that for a second. So if dad has to be homozygous recessive if it's a dominant trait. So dad is little n, little n. Mom could be big N, little n. Could that explain why 3 of her children have it and 2 of hers don't? If they inherited it, yeah. If she's heterozygous and it's a dominant trait, she could be. So there's absolutely a possibility that you would be asked to analyze a pedigree in that way. So same thing here. We don't have time to totally analyze it, but things that we noticed, recessive traits often, not always, skipped a generation. So you see them in the first generation and you see them in the third generation, but you don't see them in an in-between generation. So that's one idea. Dominant traits are often in all generations. And sex-linked, who do we see having sex-linked more often? Males. But in this weird case, it could have been any of these. These are all possible for this particular pedigree. So there's no hard and fast rule. We're just trying to help you go a little bit faster with remembering some of those little examples. Was there a question?
What if skip a generation?
Recessive sometimes skips a generation.
Oh, was the colored in one recessive?
The color. So for this one, any of them could have been possible. So recessive or dominant could have been possible. So it's just an idea, not always the case, though, not always the answer. All right, and I want you to really remember this because this is something that's so easy to forget. Not all of our DNA is carried in the nucleus. We have DNA in our mitochondria. The sperm, this is really important, sperm and pollen only have the nuclear DNA. So we're getting half of our nuclear chromosomes from dad. But the DNA that's in the mitochondria, that's from the egg. The egg is full of all of the organelles. So the mitochondria is always coming from the mom. So we don't talk about it much, but there are many, many, many mitochondrial disorders, and those are always passed through the egg. So the female plant or the female, animal, it doesn't matter, but it's always passed through the egg. Okay. Non-disjunction. Who remembers what non-disjunction can lead to that you guys all are familiar with? Down syndrome, that's correct. So non-disjunction is when the chromosomes are supposed to separate from each other during meiosis, so that half of them go to one side and half of them go to the other. But in non-disjunction, one or more of them stay stuck together, and you end up with sperm, egg, or pollen that have the wrong number of chromosomes. In a human being, that is always a genetic disorder. So examples of genetic disorders that we talked about: trisomy 21, which is Down syndrome; Klinefelter's XXY boys. So they have an extra X chromosome that leads to too much estrogen. And then Turner syndrome XO females. So that means they're missing an X chromosome. So they just have an X. That's what makes them female, because the only way to have male genitalia is with the Y chromosome. That's where it comes from. So that's why they're automatically girls when they have XO, biological female. Okay, and then polyploidy in a human being is always deadly. We saw a really crazy karyotype one time where all of the chromosomes had three. There were three of everything. That was because why? Does anybody remember? Two sperm got into the egg at the same time. Absolutely a fatal condition. Humans can barely handle one extra or one missing. Three of everything is absolutely a miscarriage. It will not survive. However, there are some organisms that do, that is their normal, is to have polyploidy. And we think that it's an advantage in the same way that females having two X chromosomes, they've got this like backup. The same thing can happen with some of these organisms. If one of their chromosomes, it doesn't matter if it's a sex chromosome, if one of their chromosomes isn't functioning, they have some extras that they can kind of count on that will work. That's not how human physiology works, but there are organisms that do better with these extra chromosomes. Okay, that is the end of heredity, I think, and now we're moving on to DNA and gene expression. How are we doing time-wise? We're doing pretty good. So just reminding you of DNA structure, we actually did talk about this in our biochemistry unit, but nucleotides on one strand are covalently bonded to each other. So if you look, and these are called phosphodiester bonds. I don't think anyone would ever ask you that, but you might see it on a test, like just they might say the word. So the left side and the right side, they're covalently bonded and they are super strong. But the nitrogen bonds across, I mean, the nitrogen bases across from each other, they are hydrogen bonded, which a hydrogen bond is strong or weak? Weak. So the two sides can separate from each other really easily. And that's part of how we can replicate DNA, is that we can separate the two strands from each other. So, and remember, the two strands are called anti-parallel, which means they're not really like this. They're like what? Nice. Upside down from each other. It's hard to do that on the video. Good. Okay. And then I don't think anyone would test you on this, but just being aware of it. The ones that have two rings, look at the diagram, just let your eyeballs see it. You don't need to know the words, but let your eyeballs see it. The bases that have two rings, these two have two rings, they're called purines. And the ones that only have one ring are called pyrimidines. Notice, I'm going to go back for a second, that you always have a one ring across from a two ring. Here's a two ring across from a one ring. So there's always three nitrogen base like rings going across. So what would happen if you had a mutation that put a T across from a C? What would happen to the DNA itself? It would be a little, not too fat, too skinny right there. It would go in, and vice versa. If you had two of the twos on the outside, you would have a bulge in the shape of the DNA there. And then remember, we have uracil instead of thymine in RNA only. They think uracil takes a little less energy to build, so we use it for RNA, the messages.
What would happen if there is it possible for what if there's like a mutation where it's like the guanine across from the thymine? What would happen there? Because it's purine and pyrimidine.
So it still is a mutation. So it's going to cause the DNA looks beautiful still, but it's could, depending on where we're getting to this, depending on where the codon is, where that mutation is on the codon, it might cause no impact whatsoever, or it might cause a change in the amino acid, which again may cause nothing or may cause something like cystic fibrosis. One letter change can change that one amino acid and can cause a genetic disorder. It varies how important that one is. Okay, replication. What does to replicate mean? So copy. Okay, so this is how we're copying the DNA. When does replication happen? Pull it all together. This big picture. When is this actually occurring? S phase of interphase, so before mitosis. It happens before the cell divides. So we're going from a monovalent chromosome to building a bivalent chromosome. So that's what replication is. It's semi-conservative. What does that mean? Okay, one old and one new. So you have the old strand is on the back, the dark blue on the diagram is the old strand, and then the light blue is the new strand. So you're always conserving the original strand, which is very helpful when it comes to mutations. Okay, and then I was going to say one more thing about it. I can't remember. 5' to 3'. Yeah, there was one other thing I was going to quiz you on. I can't remember. So topoisomerase is what's untwisting. Helicase is what's unzipping. They would never say what unzips. At least I don't think they would. But they might say what's wrong with this image, and they might mess around some enzyme or something like that. And so just be aware of that's what's going on. The RNA primers. Do they show a primer? Yeah, they have an RNA primer right here. The primers show DNA polymerase where to begin replication. So it's just giving them sort of instructions, start here. And then the RNA primers get removed. The leading strand is the one that's only in this diagram, happens to be on top. It's not, remember these are three-dimensional molecules that can be drawn in any direction. In this diagram, the leading strand's on top and it's continuous. The lagging strand forms these Okazaki fragments. And then they have to be put together using ligase. Ligase makes a covalent bond between them, so the lagging strand is as stable as the leading strand. Okay, and then that's called the replication fork when it splits like that, when the helicase is pulling it open. So it occurs during the S phase, turns monovalent chromosomes into bivalent. After proofreading, remember that we have enzymes whose job it is to go back and fix things. So those enzymes, after they've proofread your replicated DNA, the error rate is like 1 in 10 billion. But you have about, suddenly I'm forgetting, 3 billion letters. So every few divisions you're accruing more and more mutations. And so that's why it says it right there. I didn't ask it in time. Why cancer rates increase as we get older is that we're adding more and more mutations every time that go kind of slip by unnoticed. All right. So remember that what DNA does is it makes a message. It doesn't make a message, but it's the blueprint sort of. And these messages called mRNA leave the nucleus and go to a ribosome, and they tell the ribosome, this is what protein the cell wants you to build right now. The nucleus wants you to build right now. So they're kind of in charge. So the messenger RNA is what we're talking about here. So it says RNA polymerase attaches to a promoter region. This is hard. Stare at it. Be familiar with the diagram, but don't worry about being able to say it back to me. Okay? Just look at the diagram, follow along. RNA polymerase attaches to a promoter. So here's the promoter region. It's in green. And it's in front of the gene that we're going to want to transcribe. We're going to try to make mRNA out of this. Prokaryotes, the RNA polymerase binds directly to the promoter, but eukaryotes have to have these grapes. They're called transcription factors, and that helps the RNA polymerase bind more tightly and more efficiently so that it works a little bit better. And then eukaryotic promoters have a characteristic DNA sequence like a Tata box. I saw the Tata box on one AP Bio exam. It was mentioned. It's not that you needed to know what a Tata box was, but it's mentioned on it. So Tata boxes are just like a pattern of TATA that tells the RNA polymerase this is the promoter. It's time to start transcribing here. Okay, and then we need to elongate. So it reads only one side of the DNA. That's called the template strand. The other side is called the non-template, which I think is a nice, easy name, or a coding strand. So it's not being copied. The RNA nucleotides, remember, are A, U, C, and G, and the RNA polymerase just adds them one at a time until we form this long strand of RNA. And then now we need, if this is a eukaryote, we need to process the RNA because RNA has introns and exons in the eukaryotes. Prokaryotes, they don't have those introns and exons, so none of this happens. None of it also happens because the RNA isn't in a nucleus. In a eukaryote, the RNA has to escape from the nucleus. That's part of the 5' cap. But in a prokaryote, in a bacteria, it doesn't have to escape from the nucleus. So it doesn't need any processing at all. It's as is, ready to go. Okay, so a modified nucleotide is added to the 5' end. We call that the 5' cap. And then a tail of several hundred adenines is put on the 3' end tail. It's called the poly A tail. We learned that there are some types of microRNAs who eat the mRNA from the tail end. They eat. And once they eat the poly A tail, then the mRNA doesn't work anymore. And so, in essence, you've shut the gene off. We'll get to that. But just they eat the poly A tail. Okay, introns are removed and exons are spliced together. I feel like those two words are backwards. Does anybody else feel that way? I feel like exons should be out, and that's not what happens. The introns get taken out. The exons get put together, and alternative splicing says that the exons can be put together in different orders. And so that means that one gene can code for several different proteins depending on how those exons are put together. It's really remarkable. If you are like, I don't get that, that's okay because it's incredibly remarkable how the cell knows how to do that. Okay, and then a quick reminder, ribosomes have a small subunit and a large subunit, and they're the ones that are going to build the proteins. So there's 3 sites, the A, P, and E, they go kind of backwards sites. So the A site is where the amino acids enter, the P site is where the growing polypeptide is kept, and the E site is where the empty tRNA molecules leave. I will show you that. It'll make sense in a moment. So these are the trucks. I call them trucks, the tRNAs, because they carry an amino acid. Look where the diagram says it. The amino acid is carried up at the top, and then down at the bottom are the 3 letters that are called the anticodon. That is because they are the opposite 3 letters of the mRNA codon. So the code is on the mRNA. So if you have AAA on the mRNA, what will the truck's anticodon say? What goes across from A? U. Oh, right. No T's. So this is going to be U's. Very tricky. Okay, moving on. So you need to be able to read either of these tables. I always felt like this table, the round one, is quite intimidating. The first letter is in the middle. So for example, let's look up AAA. Let's find AAA. So here's the first letter, here's the second letter, here's the third letter, and you've got it, okay? You can also find it on this table, where are the A's, sorry. A down here, second A here, there we go. And lysine is right there, okay? So a couple things, 3 letters on the mRNA is a codon. There are 64 possible codons because you've got 4 letters and 3 different positions for those 4 letters. So more than one codon can code for a particular amino acid. So UUU and UUC both code for phenylalanine. What does anybody remember what it's called? I don't think they'll test you on this, but if you mutate the first or the second letter, it doesn't have any impact. What is that called? It's got a funny word. It's wobble. That's wobble. And that's nope. So wobble describes the redundancy in the genetic code. The same amino acid may be encoded by multiple codons. The third letter is actually the important letter. The first two letters are not important. It's the third letter that determines which amino acid it is. Crazy, right? And then remember that there are a few stop signs. So a few of the codons stop it. And then there's one start, and that's the AUG. It is also methionine, but that's the start codon. Okay, moving on. Is there anything on there? I don't know about that. Okay, translation. I always hate the words transcription and translation. Transcription is making RNA. Translation is making a protein. So take a look at the image from beginning to end. We've got DNA. It's split apart. The RNA polymerase copied one side. So here's our pre-mRNA. We cut out the introns. We add a poly A cap. I'm sorry, a 5' cap and a poly A tail. It leaves the nucleus. A ribosome clamps onto it. Small subunit, large subunit. The trucks start pulling up, and they start dropping off their amino acids. And as they drop off their amino acids, we start building a protein. And what's a baby protein called? A polypeptide. So until the protein is really built, we just call it a polypeptide. Okay, that's everything. We'll stop when we get to a stop codon. Okay, translation in prokaryotes versus eukaryotes. Since prokaryotes don't have a nucleus, translation can be coupled with transcription. So remember, this is called a polyribosome. So as fast as the DNA is being read, here's your mRNA being made, the ribosomes immediately clamp on and start making proteins. That's super, super fast. Eukaryotes are way slower, but we have much more control. So there's places where we can stop it and restart it and stop it. We have all this control, whereas bacteria, they just don't control anything. It's just super efficient, but there's no turning things on and off like we can do. Okay, retroviruses, just a quick reminder, HIV is a retrovirus. Look at this and just make yourself go, oh, that's not right. They have RNA instead of having DNA. So the RNA gets converted into DNA, and then the DNA makes the RNA, and then that makes a protein. And the problem with retroviruses is the enzyme that's making the DNA from the RNA is called reverse transcriptase, and nobody proofreads it. It's terrible at its job, and it makes constant mutations. So HIV is constantly changing. I've been told that pretty much every patient that has HIV has their own variety of HIV, and that's why we're having such a hard time making a good vaccine against all of it. We've got good medications to stop it once somebody is infected, but we don't have a good preventative in the first place. So preventing people from getting it. Okay, and then gene regulation. I'm not going to go through a whole lot of details on this, thankfully, right? But just the idea that we can stop or turn off a gene in order to conserve energy. So the example that we had was like if we're digesting lactose, if we haven't eaten any lactose, we don't want to be making the gene or making the protein for digesting lactose, because that would be a waste of energy, right? So we only want to produce the protein for digesting lactose when we've actually eaten lactose. And so that's gene regulation. If all of our genes were on all of the time, we would immediately run out of energy. We can't survive that way. We don't have enough energy stored in our bodies for all of our DNA to be triggered all at the same time. Okay, so let's back up and talk about the operons, and I know Sammy, you were asking about this. So we learned about two types of operons: the trip operon and the lac operon. So it says these are organized clusters of genes that contribute to a particular metabolic task. There are inducible and repressible. If you induce it, like inducing labor, you're triggering it. So it's usually off, but we can trigger it. Repressible means you can stop it. So it's usually on, but we can stop it if we need to. Or the bacteria. These are not in humans, these are in bacteria. They are both examples of negative gene regulation because there are repressor proteins that shut them down. So here's the lac operon. It's inducible, which means it's normally shut off, but the bacteria can activate it when there's lactose present. So notice the repressor. Look at the diagram. I don't need you to memorize a million words. Look at the diagram to see if you can understand it. So this whole thing, remember, this is DNA. It doesn't look like DNA, but they're showing the double helix right here. This is all DNA. And the repressor is sitting on the DNA. It's actually physically attached to the DNA, and the RNA polymerase can't get in there because the repressor protein is bound to the DNA. It's in the way. So right now we say that the lac operon is shut off. There's no lactose present. We don't want, the bacteria doesn't want it to make the enzymes for digesting lactose because that would be wasteful. Now, in this diagram, notice that lactose is present. The lactose molecule, where is my cursor? There it is. The lactose molecule right here is the green circle. It's going to touch the repressor and make it change shape so that it can't hold on to the DNA. And so now the RNA polymerase can move across. So it's going to read the genes. It's making the mRNA. Here's the mRNA. And that is eventually the trucks are going to pull up, all that's going to happen. And that's going to lead to the formation of the 3 enzymes that help this bacteria digest lactose efficiently. The opposite, to say it's, I'm sorry, there's one more thing about this. If the cell wants this to happen super fast, then there's also this thing that we joke was kind of like it turbocharging it. So when lactose is present and glucose is low, the cell is hungry. Low glucose means the amount of cyclic AMP is high. So this orange diamond here, if there's a lot of that, it's going to trigger what is called CAP. So the catabolite activator protein. It'll attach to the RNA polymerase and make it work faster. So we've been talking about turning the gene off. This is actually turning the gene on to make it go faster. This is causing the RNA polymerase to go even faster, so it'll digest a little bit faster. Okay, just a quick reminder about small RNAs. There are a bajillion different types of small RNAs. They have lots of different names: microRNA, small RNA, siRNA, RNAi. Don't panic when you see that. They're all just small. And their main idea is that they can either degrade. So this example right here, it's degrading the mRNA so that we don't make that protein anymore. So remember, the mRNA has left and it's gone to a ribosome. The ribosomes can read it over and over and over and over again, and they're going to make lots and lots of the enzyme until somebody makes it go away. The microRNAs can cut it up into pieces, make it go away, or they can just snap on and get in the way so that the ribosome can't attach to it anymore because they're in the way. So they're physically in the way. The ribosome can't read it. So again, we're not going to make any more proteins. So they either degrade the target mRNA or they stop translation. And so we're not going to make any more of that enzyme. Either way, the target protein is no longer produced. Okay, let's review mutations here really quickly. 2 major types of mutations: point mutations or frame shift mutations. Some mutations theoretically are beneficial. That's the idea of evolution, right? Sometimes a bacteria has a mutation that makes it immune or resistant to an antibiotic. But most of the time, mutations are either not going to affect us at all or they're going to be harmful. It's a pretty rare thing where we would see a mutation that actually gives an organism like us an advantage over others. Maybe disease resistance would be an example of an advantageous mutation in an organism like us. Okay, so then a point mutation, just one letter is changed. So look on the diagram here, make sure you see it. So GAA gets changed to GUA, and now we have a new amino acid that gets put on there. And so that's called a point mutation. And then frame shift mutation is, give me a second. Oh, we don't show it. Frame shift mutation is when you delete or add. And so all of the groupings of 3 after that are screwed up. Do you guys remember that? Feel good? Okay, can I move on? Okay. How do prokaryotes evolve? Prokaryotes clone themselves. Bacteria clone themselves. So how are they evolving? So when they reproduce, there's several things that can happen that allow them to evolve, not revolve. They can be transformed, and that is such a lame vocabulary word because it sounds like I don't know what it means. Transformation means, remember, it means that they can suck up the DNA from the environment. So if one bacteria just happens to die, and it's like we joked, if I got hit by a car and Jamie could walk up to me and go and suck up my DNA, and suddenly Jamie is a hybrid between me and them. That's what bacteria can do. Bacteria can suck up the DNA, and suddenly they have that DNA. They've magically transformed. We can do nothing like that, but they can get new DNA. So this is how they evolve. They can get new DNA by sucking up the DNA from a dead bacteria. They can have bacteria sex. There's no such thing as a male and a female in bacteria, but they can trade. They have this conjugation tube, and they can get new DNA from a bacteria, not even a close relative, it can be a different species, and they can get new DNA from a completely different species. It's a very wacky thing. Transduction is when a virus does this, and it's an exceedingly rare event, but because there's so many bacteria and so many viruses, it actually happens fairly frequently, which sounds weird. But this is where a virus injects its DNA. The pink is the virus's DNA. We build new viruses. The bacteria builds new viruses, and they come bursting out. But notice this first bacteria or first virus. What's wrong with this first virus? It's got some blue in it. It's got some of the bacteria's DNA trapped inside of it. So now when that virus goes and infects a bacteria, it injects some bacterial DNA. The viral DNA is not going to work because it didn't get enough of the viral DNA. So this cell is going to survive this event. The bacteria is going to survive this event, but now it has new DNA. And look in this last diagram. The new DNA has incorporated into the bacteria's DNA. So suddenly this DNA has changed. It has evolved for better or worse. Might be a terrible thing, but it's got new DNA now. Might give it an advantage. All right, and then the last thing are called it's transposition or transposons. Some DNA can jump from one location to another. And so in bacteria, they have their main circular DNA, and then they have these little things called plasmids. And sometimes genes can jump from their main DNA to a plasmid, and then the plasmid is what gets transferred through conjugation, or maybe gets spewed out when the bacteria dies and another one sucks it up. So it's just another way. Antibiotic resistance genes are often on genes that jump. So kind of a creepy thing. Okay, and then biotechnologies, we're getting near the end, everybody. How many slides do we have left? Liam, we're pretty close, aren't we?
Yeah, I think so.
Hopefully not too many. Restriction enzymes are enzymes that cut DNA, and we use them in biotechnologies. They're super cool. We can use blunt ones, or we can use ones that create the sticky ends. You've got sticky ends on this one. And we use them either for like DNA fingerprinting, or we use them for genetic engineering experiments. Who remembers, do we need a blunt end or a sticky end for a genetic engineering experiment? sticky end, because we're going to add a new gene and we need it to stick to the other genes. We need them to be attracted to each other. So this is what you're seeing right here. We've added in this genetic experiment, we've added a new gene with the opposite letters, and it sticks to the bacterial DNA. Okay, and then PCR, remember, is just a machine that copies DNA. So if we have a blood stain, this is one example. If we have a blood stain at a crime scene, it has an incredibly tiny amount of DNA. Yes.
Why do endonuclease exist? It seems like in the wild it'd be disadvantageous.
That's a super, super good question. Give me just a second because I know the answer. There's a really good answer. The question was, why do endonucleases exist? And they exist naturally. And suddenly I'm forgetting why they are there. Oh, because they are the editors that cut out bad DNA and reattach the good DNA. So they exist correctly to edit DNA when DNA has a mistake in it. And we've learned how to use that. Okay, super good question. PCR, remember there's a tiny amount of DNA in this drop. PCR copies the DNA, and here's how it copies it. So we denature it by adding a lot of heat. The DNA comes apart. Then we add these primers that keep it from coming back together again. And then we add a special type of polymerase that copies the DNA. And then we repeat that process over and over and over and over and over. And within like an hour, we can have a test tube full of that person's DNA. And now we can run a DNA fingerprint and figure out whose blood that was, which is really, really cool. Or if it's a gene of interest, let's say we've been working for 20 years to isolate the gene for insulin or the gene for human growth hormone, and we've got this one copy of it, we throw it in a PCR machine, and an hour later we have 1 billion copies of it. That's really great. Now we can work with it. Now we have something we can do with it. Okay, DNA fingerprinting, just a quick reminder. The chunks of DNA, are they positive or negatively charged? They're negatively charged. So when we turn on the electricity, they push away from the negative end and they move toward the positive end. Which ones travel further, the big ones or the little ones? The small ones experience the least resistance. They travel the furthest across. And so everybody produces their own unique DNA fingerprint. And so, whoops, hold on. So here's my example where I showed that we cut the DNA, and the DNA didn't get cut right here because there's a change in the DNA. So it produces a different fingerprint. Each person has a different fingerprint. All right, and then sorry, I was going to not tell you the answer to this one. We have a child. This is their mother. And then which one is their father? DNA fingerprinting can be used for lots of things, including paternity. When it's paternity, it's half of the bands come from one parent and half of the bands come from the other parent. So which parent is the parent of the child? Parent number, they're both males. Do y'all see that? Male number 1 and male number 2. Good. All right. You can practice others if you want. Here's how you do a genetic engineering experiment. Cut it with a restriction enzyme that produces sticky ends. Add in your gene of interest, and then treat it with an antibiotic that kills the bacteria that did not absorb your plasmid that has been genetically engineered. And then all of the bacteria start mass producing this protein. And so if that protein is insulin or human growth hormone or whatever, now you can milk the bacteria, like you're milking a cow or something. You can milk the bacteria for this particular product that they're doing, which is really cool. Examples of recombinant DNA. We've made genes for pest resistance that we've added to plants. We've added genes to bacteria to clean up toxic waste spills. Cool. So smart. We've added proteins to dissolve blood clots, like if somebody's got a blood clot in their leg, we can do that. And human growth hormone or whatever to folks that don't have normal height for some reason. Last one, I think. Is this last one? Second to last one. Okay. DNA sequencing, the Human Genome Project was the one that determined the order of all the A's, T's, C's, and G's in the human genome. We don't know what they mean. We know the A's, T's, C's, and G's, but we don't know what all those A's, T's, C's, and G's mean. So I can't tell you every gene. We can just tell you all the letters. Human beings are like 99.99% the same letters. So while you think you're so unique, you're not that unique. So we know most of all of your letters in a row. And then this colorful thing, this is called the Sanger method. And it's just a way of analyzing the A's, T's, C's, and G's. Each color tells us a different letter. So that's what this colorful picture is. So like the reds would be the T's and the greens would be the A's and the whatever. And so that's what that's telling us is the DNA. Okay, that's it, everybody. Did I get it a little bit over an hour, I think? But I tried awfully hard. You guys go enjoy your Friday. I hope everybody has a nice day later.